Hi everyone, my name is Annalena and I'm a PhD student at the Laboratory for Brain-Gut Axis Studies in KU Leuven. So today I will be speaking about the gut-brain axis. So in my PhD, I'm actually investigating the connection between the gut microbiota and stress sensitivity. So my main project is actually focused on which bacteria are linked to stress, but another project is also on how these bacteria affect the brain and how they communicate with the brain. And that's what my talk will be about today. So in our lab, the main mediator of interest that we're investigating are short-chain fatty acids. So they are derived from dietary fiber, which bypasses the digestive organs and actually serves as food for the bacteria. So there are specific bacteria that ferment the dietary fiber into short-chain fatty acids. And acetate, butyrate, and propionate are the most abundant short-chain fatty acids in the human body. And actually from the colon, they can already indirectly interact with the brain via vagal pathways, humoral pathways, and inflammatory, the immune system. But a fraction of the short-chain fatty acids also reaches the systemic circulation, where we can measure them in blood. And they have even been shown to cross the blood-brain barrier and have been measured in the brain in animals. So that means they can potentially affect the brain directly. So the evidence from animal research shows that short-chain fatty acids have a beneficial effect on stress-related behavior and stress physiology. And in our lab, we want to investigate this in humans. So first of all, we were interested in understanding how short-chain fatty acids are related to the stress response. So to investigate this, I used data from 3 studies on healthy young males that all underwent the same study procedures. So they went on a 3-day low-fiber diet to standardize the fiber intake, and they had to collect a stool sample. Then on the day of the study visit, they collected saliva samples in the morning to quantify the cortisol awakening response. And at the study visit, we performed the Maastricht acute stress test, which is an experimental stressor that induces a robust cortisol response. So we measured that in saliva, and we also measured self-reported stress levels, and we collected blood to quantify the serum short-chain fatty acids. So here are the results for the cortisol awakening response. So we use short-chain fatty acids to predict the cortisol concentration, and serum butyrate, serum propionate, and total serum short-chain fatty acids negatively predicted the cortisol concentration. So that means that the higher the concentration of that short-chain fatty acid was in the blood, the lower cortisol was. And here are the results for the cortisol reactivity to the stress task, the MAST. And here, serum propionate and serum total short-chain fatty acids both significantly predicted cortisol reactivity, again in the same direction, so negatively, meaning that the higher those levels were in your blood, the lower cortisol reactivity was. And even though acetate and butyrate are not significant, they are in the same direction, so that is nice to see. And these results are actually complementary to a study that my supervisor performed, where she studied the mechanistic role of short-chain fatty acids. So she performed a short-chain fatty acid intervention. So she actually delivered short-chain fatty acids directly to the colon, where they would naturally be produced using colon delivery capsules. And then she randomized 66 healthy young males to 3 different groups. A low dose of short-chain fatty acids, which would correspond to the ingestion of about 10 grams of fiber, a higher dose of short-chain fatty acids corresponding to about 20 grams of fiber, and placebo. So this is the design of the study. She had them go on a low-fiber diet. There was a pre-intervention visit, a post-intervention visit, which was separated by 1 week of the intervention. And she found that both doses of short-chain fatty acids actually reduced cortisol reactivity to the stress task in comparison to the placebo. And importantly, the differences in the serum short-chain fatty acids co-varied with the changes in cortisol. So that means the more the short-chain fatty acids increased in serum from visit 1 to visit 2, the greater the reduction in cortisol was. So that means, this is quite cool, because that means that the short-chain fatty acids should be in the serum to kind of give this effect of reducing cortisol. So that means there could be a direct effect of short-chain fatty acids on the brain. So the only thing is that we don't really know the mechanism yet, and that's what I'm going to be investigating in my next study. So I will look at what's going on in the brain. I'm going to use functional magnetic resonance imaging and the Montreal imaging stress task to see the effect of these short-chain fatty acids on brain activity. And I'll be focusing on brain regions that are associated with emotional processing of stress and cortisol release. And I also want to investigate the hypothesized mechanism of action. So from animal research, it seems that short-chain fatty acids modulate epigenetics. So a very quick recap of epigenetics. Basically, the DNA is wrapped around these histones, and that forms the chromatin structure. And if the histones are acetylated, then the chromatin structure is more relaxed. That means the transcriptional machinery can access the DNA and gene expression can occur. Histone deacetylases, on the other hand, they remove these acetyl groups, and then there's the DNA is wrapped more tightly around the histones, and the transcriptional machinery can't really reach the DNA, and that means gene expression is reduced. So this is actually what is observed in animals that undergo a lot of stress, that they have more expression of HDACs. And histone acetyl transferases, they do the opposite action, so they add acetyl to these histones, and then this enables gene expression of genes that are important for neurogenesis, synaptic plasticity, BDNF, and so on. And HDAC inhibitors also show this effect, and that's the hypothesized mechanism of action. Short-chain fatty acids are HDAC inhibitors, so that's from animal research. This is what is suggested, but it hasn't been shown in vivo yet in humans, so I'm going to be doing that. So I will be using positron emission tomography with a radiotracer carbon-11 martinostat. So this is a selective imaging probe that for HDAC, specifically the isoforms that are related to stress and short-chain fatty acids. And yes, my study will start hopefully next month. So I will replicate the study that my supervisor did and include these neural measures. So I want to investigate the effect of 1 week short-chain fatty acid intervention on HDAC expression in the brain, neural responses to acute stress. I also want to see whether, well, if they have effects on the brain, whether they mediate the effect on the cortisol response. And lastly, I'm going to add some measures that weren't included in the first study, which are also likely to be influenced by short-chain fatty acids. So that's inflammatory responses to stress and the autonomic nervous system response to stress. So I will recruit 32 healthy males and will randomize them to the colon-delivered short-chain fatty acids or placebo. And the study design will be very similar. So they will undergo a low-fiber diet, pre-intervention visit, followed by a post-intervention visit with 1 week of intervention in between. This is the study visit. So we'll perform a PET-MR scan with the Montreal imaging stress task in there. We also still have to do the mast acute stress task again, because that induces a robust cortisol response, which the MIST doesn't do. And we'll add the ECG to the MAST and some blood pressure measurements as well. So the implications of this study: first, we'll gain valuable evidence, in vivo human evidence, on the hypothesized mechanism of action of short-chain fatty acids. And we'll also gain more knowledge on the effects of short-chain fatty acids on stress responses and possibly how they modulate cortisol reactivity. This is then important for the future when we want to look at clinical populations with stress-related disorders and also at nutritional intervention that target short-chain fatty acid levels. And then lastly, also methodological implications. So basically, this radiotracer has only been used in case control studies, so we'll be the first to use it to evaluate an intervention on the brain. So yeah, we can potentially validate it for pharmacodynamic brain imaging, and that's then really important because there's a lot of neuropsychiatric disorders that are related to HDAC dysregulation. This is my lab, and yeah, thank you very much for paying attention.
So, questions for Annalena? Anyone? I do have a question. So very naive question. How would you link these results you've shown with a ketogenic diet that is really a lot has been done, a lot of pseudoscience behind, but recently has been said like short-term ketogenic diet is beneficial, but long-term might be not so beneficial. So
What kind of diet?
this ketogenic, like high-fat diet.
Okay, I
Some
don't
of
really
it.
know about
I don't know. Okay.
about
Okay.
high fat. So I
Healthy fat. Yeah.
yes. So I can't say that much about it because short-chain fatty acids aren't really linked to that. But I
Okay,
can
okay.
say that we tried the same study just with fiber and wanted to look at cortisol reactivity.
Yeah.
But we only tried one type of fiber and we didn't see it affect cortisol reactivity. But that doesn't mean much because the reasoning is probably that short-chain fatty acids need to be increased in a certain amount to have that effect. And that means we would have to find a fiber intervention that would achieve the short-chain fatty acids to reach those levels.
Okay.
So I'm not sure about
Yeah, yeah.
fat in
Yeah,
that
yeah, yeah.
context.
Thank you.
Thank you very much for the very interesting talk. I have two very naive questions. First, can you remind me again which is the macronutrient composition that will bring the short-chain fatty acids? And also this colon delivery capsule is like a capsule that will only break down at the level of the colon? How does it actually work?
Sorry, I didn't understand.
Once again, first question is, which is the macronutrient that is actually transformed into short-chain fatty acids?
Which bacteria?
Which macronutrient? Is it
Macronutrient. Oh, it's fibers. There's
fiber?
different types of
Okay, that's what I thought.
fibers. Yeah.
And how does this colon-directed delivery capsule works?
Okay, so they're coded in a specific way because the pH levels change throughout the gastrointestinal tract, and then they only open when the pH reaches, I'm not really sure what it is right now, but then, so we have tests to check that they actually open only in the colon and not before.
Okay, thank you.
Yeah. Last question.
I was wondering if next to the cortisol measures, you also had more subjective measures of whether...
Oh, yes. Sorry, I forgot to mention it. Nothing was significant for the subjective measures to stress, but we have to say that these are healthy young males, and my supervisor investigated that the physiological responses don't correspond to the subjective responses. Maybe in women it's different. Yeah, but for now we don't see an effect there.
Right, because I saw the diaries, so I was wondering if you were just, collecting information on what they were eating, or also sometimes they collect like the emotional responses to the food they are intaking or how they are feeling during the day, and all these kind of things that could tell you something about the mental state of the person and how it's affected by the food.
Yes, we do collect data like that, but it's the only intervention is the short-chain fatty acids here. Everything else is pretty much controlled, and there we don't see effects.